In this video, we're going to learn about a number of cellular components and their functions. The simplest type of cell there is are prokaryotic cells, which compared to eukaryotic cells, as I mentioned, are pretty simple. A prokaryotic cell has a number of basic components. So the first thing that we're going to talk about is the cell membrane. All cells have a cell membrane, so does bacterial cells. That's the plasma membrane that encapsulates the cell and keeps the components inside the cell. Bacterial cells also have a cell wall. It's a component of the cell that is mainly made of carbohydrates, and it helps make the cells rigid and help them with survival in the outside environments that they live. You see another more thicker covering, which is called capsule. This is a component, a covering that you would only see in pathogenic bacteria, which are more rigorous and they survive better. So that's what the protection that the capsule provides for them. But not all bacterial cells have this capsule. However, all bacterial cells have a cell membrane and a cell wall. Then bacterial cells have, just like any other cell, they have a genetic material, and the genetic material of a bacterial cell is one piece which is circular. You cannot see that it's circular in here because it's kind of have shown in its wiggled shape, but if you were going to draw it, the bacterial cell DNA is just one circular DNA. These dots that you see inside are ribosomes. These are cell structures that are involved in protein synthesis, and pretty much all cells have ribosomes in it. So now this is a good time to talk about components that all cells have. So one of them is genetic material. So we're kind of DNA. The genetic material. All cells have cell membrane. This is the plasma membrane. Not all cells have cell wall, so be careful. Cell membrane is not the same thing as cell wall. All cells have cell membrane. All cells have ribosomes. And also, all cells have this soup inside of them, inside in here, which is called cytoplasm. This is just the space inside the cell where all the components are sitting. So all cells, no matter their origin, they have DNA, they have a plasma membrane, they have ribosomes, and they have a cytoplasm. Oh, another thing that I didn't mention with prokaryotic cells is this structure in here. It's called flagellum. Bacterial cells do live in solutions, but they also can move around. So this flagellum is a structure. It's kind of part of the what's called cytoskeleton part of the bacteria that allows it to move. Sperm cells also have, so animal sperm cells also have these flagella which allow them to move. These pili are protrusions from the bacterial cell which allow different bacterial cells to exchange genetic material with one another. Now, compared to prokaryotic cells, eukaryotic cells are pretty complex. They have a lot of membrane-bound organelles in them, each of them having their own functions, but prokaryotic cells do not have these membrane-bound organelles. On the top, you see an artist's representation of an animal cell. On the bottom, you will see a representation of a plant cell. Pretty much most eukaryotic cells have the same kind of organelles, but with some exceptions. Now this is a video. You have access to the slides, so there's a video linked behind this image which talks about the different components of a cell. It gives you a nice representation, visual representation of all these things, so I invite you to watch this video. So this is a representation of a eukaryotic animal cell, and we are going to talk about all these different parts of an animal cell. You are expected to know all of these components, their names, and their function. So let's begin with the plasma membrane, which is this barrier that is around the cell. That's the phospholipid bilayer. These are these red structures with legs on them. These are the phospholipids, and they are forming a bilayer. Now, a cell's plasma membrane isn't only phospholipids. It has a lot of different things in it. It has proteins that are embedded into the plasma membrane, and also proteins that are sitting on either side, either inside or outside the cell. The proteins that span across the membrane, they're called integral membrane proteins. Some of these integral membrane proteins have openings inside them such that they provide a pathway for some molecules to pass into and out of the cell. In that case, we call them a protein channel. The proteins that are not embedded into the plasma membrane, but they're sitting on either side, they're called peripheral membrane proteins. Then, in addition, within the plasma membrane, you see these molecules of cholesterol. Usually when we hear cholesterol, we think that it's an evil molecule, but just like anything else, if you have too much of it, it's not good. But actually, cholesterol is a very important component of our plasma membrane. It gives it integrity. We will talk about this more when we discuss the plasma membrane in more detail. Other things that are in the plasma membrane are these carbohydrates that are attached to either the proteins that are sitting in the plasma membrane, or they're attached to the phospholipids. If the glycoprotein is attached to the protein, we call this entire thing a glycoprotein. And when the carbohydrate is attached to the lipid, we call it glycolipid. And usually these glycolipids, and specifically the carbohydrate part of them, they're involved in cell-to-cell interaction. So there are a number of very important functions that the plasma membrane provides. One is that it forms a boundary around the cell, which separates its internal components from the outside. The plasma membrane very tightly controls what goes inside the cell, so it controls the traffic of molecules across the plasma membrane. It is also involved in mediating cell-to-cell interactions through these proteins that are in here, and also through these carbohydrates. And also it receives signals and passes signals from the outside to the inside. And again, very often these signals are passed and received through these proteins that are sitting inside the plasma membrane. Sometimes the signals are passed through these phospholipids as well. Next, we go to the nucleus, which is a command center of the cell. Why do we say that? Because the nucleus contains the DNA, the genetic material, and all the information that is needed for a cell to be what it is encoded in its DNA. Now you notice that you don't see the word DNA here, because DNA inside animal cells is wrapped around protein, and collectively we call it a chromatin. So then chromatin is DNA plus protein. These fibers that you're seeing here is actually genetic material that is wrapped around protein. Now, the nucleus has two phospholipid bilayers, which collectively we call the nuclear envelope. So where are the two layers? There's one layer inside here, and then the second bilayer is on the outside. So collectively they're called the nuclear envelope. And the nuclear envelope has pores in it, which we call the nuclear pore. This nuclear pore performs a very important function for talk between the nucleus and the rest of the cell. So the nuclear pore very tightly controls what goes in and outside of the cell. There are actually specific signals that determines what can go in and out. So it's not like this is open and anything just leak out and in. The nucleoplasm is basically the space inside the nucleus. And you notice that the outer layer of the nuclear envelope is continuous with another structure, which is called the endoplasmic reticulum. So these are basically continuous in here. We're going to talk about endoplasmic reticulum shortly, and you see that this endoplasmic reticulum is studded with these red dots. These red dots are actually ribosomes. Ribosomes are complex molecules that are involved in protein synthesis. Ribosomes, they usually perform their function outside of the nucleus, but interestingly, they are first synthesized inside the nucleus in a structure in the nucleus called the nucleolus. So the function of the nucleolus is synthesis of these ribosomes. These ribosomes are made and packaged, and then they're shipped out of the nucleus into the cytoplasm. Which brings us to what are ribosomes? As I mentioned, ribosomes are protein complexes that are involved in making protein inside the cell. And when you look at a cell, you find ribosomes in two different areas, either floating around in the cytoplasm freely or bound to the endoplasmic reticulum. Now, these two different sets of ribosomes are synthesizing two different classes of protein. The ribosomes that are in the cytoplasm are synthesizing proteins that are soluble. They're in the cytoplasm, and they're destined to do something in the cytoplasm. But the proteins that are made on ribosomes that are attached to the endoplasmic reticulum tend to be proteins that are either membrane bound, or they carry signals that make them destined to go either to another organelle or to go to the plasma membrane on the surface of the plasma membrane, or to be traveling outside of the cell. So these proteins usually have signals in them, sequences in them that determines where they're going to go. But they're all made with ribosomes that are sitting on the endoplasmic reticulum. So here's a kind of an artist's rendition of ribosomes. Ribosome has a large subunit. This is the large subunit, and a small subunit. Everything else here is not the ribosomes. It's different things. So what happens is that the large and the small subunits of the ribosome come together with a messenger RNA that has a genetic code in it, and then with the help of another RNA, which is called transfer RNA, they all work together to make protein. You're going to learn a lot more about this later on in this course. Don't worry about it. Just remember that ribosomes, their function is to make protein. They're involved in protein synthesis, and that they're made of a small and a large subunit. We talked about the endoplasmic reticulum, that membrane that is continuous with the nuclear outer membrane. turns out that there's two different types of endoplasmic reticulum. We have rough ER or smooth ER, and each of them have their own specific functions. The rough ER, the reason they call it rough ER, because scientists, when they looked at it, they kind of look like they're rough looking because they were studded with ribosomes. So the fact that they are studded with ribosomes suggests correctly that the rough endoplasmic reticulum is involved in synthesis of proteins. And as we mentioned, these proteins are a specific class of proteins. So the rough ER is involved in synthesis, modification, and transport of proteins, which could be membrane proteins, secretory proteins. These are proteins that are secreted outside of the cell. They could be digestive enzymes, which end up going to another organelle called the lysosome, or any other type of protein that have a signal attached to them that tells the cell, ship this protein to this particular location. So that's the function of the rough ER. The smooth ER, on the other hand, does not have studded ribosomes on it, which implies correctly that it is not involved in protein synthesis. Smooth ER performs many other functions that are very important, and its function, it varies depending on the type of cell. So in most cells, smooth ER, they're involved in synthesis of lipids and phospholipids that make the membranes and all these membranes, and also in cells that make up the hormone producing cells, they're involved in production of steroid hormones from cholesterol. In muscle cells, smooth ER acts as a place for storage of calcium. So it holds calcium, and when your muscle receives a signal, it releases that calcium, which helps the muscle to contract. In liver cells, smooth ER is involved in detoxification of poisons such as alcohol or any other molecules that the cell considers poisonous. So it detoxifies them. And in liver cells, the amount of smooth ER is really, really large. Then we're going to talk about Golgi apparatus, which is in this part of the image. And the Golgi apparatus, you can think of it as the equivalent of a FedEx packaging center. And it works very closely with the rough endoplasmic reticulum. So the Golgi apparatus is involved in receiving the proteins that are made and modified in endoplasmic reticulum, and then in help modifying them and then transporting them to where they're supposed to go. And these are the same type of proteins that are made in the endoplasmic reticulum. So the Golgi apparatus constantly is talking to the endoplasmic reticulum. And how does the talking work? Using these little round structures called transport vesicles. You can think of these as the FedEx trucks. So the packages are shipped from the endoplasmic reticulum. In this case, we're looking at a transmembrane protein. So it is shipped into these trucks, and then the Golgi apparatus receives it on one end, which is called the cis face of the endoplasmic reticulum. Cis means same. Cis means same. So that means it is the side of Golgi apparatus that is closest to the endoplasmic reticulum. It is on the same part of the cell that is same side of the cell in which rough ER exists. So the cis end of the Golgi apparatus is the receiving end. It receives vesicles from rough endoplasmic reticulum. Then these proteins are modified as they pass through the different levels of the Golgi membrane. Golgi is also a double membrane organelle. And as these protein moves around, sometimes Golgi helps these proteins fold, sometimes it modifies them, adds carbohydrates to them. And then by the time it's finished, it comes out the other end, which is called the trans phase. Trans means different. So then this protein comes out using the membrane on the trans phase of the endoplasmic reticulum, buds out, forms a little vesicle, and then this vesicle moves where it's supposed to go in the cell. In this case, we're looking at the plasma membrane protein. So it goes in there, this vesicle fuses with the plasma membrane and delivers this protein. I'm going to talk about that a little bit more later. So the function of Golgi is modification and transport of a certain class of proteins. So you can think of it as the FedEx or UPS center of the cell. Here's an electron micrograph of a Golgi apparatus. This is the cis phase and that's the trans phase. And you can see all these vesicles that are budding out of the trans phase and taking stuff away from Golgi apparatus, and these are vesicles that are fusing with the cis phase. The next organelles that we're going to talk about are lysosomes, vesicles, and vacuoles. In animal cells, vacuoles don't really play a large role, but in general, they play a role as the organelle that is the bag that is holding stuff. Sometimes it holds liquids, sometimes it holds, it's a temporary location for toxic compounds that are supposed to be, that the cell needs to get rid of. Vesicles, you already met vesicles in here. You saw a whole bunch of vesicles that what it does, it helps move molecules around inside the cell. Then lysosomes are, you can think of them as the stomach of the cell. The pH in lysosome is really, really low, and it's full packed with digestive enzymes. So you can think of, again, lysosomes as the stomach of the cell. It helps digest anything that the cell wants to digest. And one example of doing this is in macrophages. Macrophages are white blood cells that are involved in engulfing, finding bacterium, engulfing them, and protecting you from infection. And this is what's happening. Usually a macrophage engulfs a bacterium through a process of endocytosis. Basically, it's a pinching of the plasma membrane inside, and this forms a vesicle carrying the bacterium. Then what happens, this vesicle comes and fuses with lysosome. Remember the bag with a lot of digestive enzymes. It digests. When they fuse, the enzymes in here digest the bacteria into pieces and basically kills it and protects us from it. Peroxisomes are these little organelles that are very important in a number of aspects. First of all, they break down fatty acids and amino acids, so they have a lot of digestive enzymes in there. They are also involved in detoxifying poisons, including alcohol and other poisons, and usually one byproduct of detoxification is hydrogen peroxide, and the fact that all of this digestion is happening in this bag, in this membrane-bound organelle, is a good thing, because then it keeps the harmful hydrogen peroxide that is a byproduct of detoxification from messing up the functions inside the cell.